Hello everyone, welcome back to the NCBI Master Series. So in previous video we explored nucleotide database, gene database and BLAST. So today we will learn one of the most important resource used by molecular biologists and also microbiologists, bioinformaticians worldwide, and that is the NCBI Protein Database. So if you want to find protein sequences, study protein functions or identify conserved domains, or if you want to download any FASTA file or perform protein analysis, so this tutorial is for you. By the end of this video, you will be able to confidently use the NCBI protein database for your own research projects. So before using any database, we should understand its purpose. So the NCBI protein database is a repository of amino acid sequences which are collected from different sources, and these sequences come from gene bank translations, then RepSeq proteins, UniProt KB, SwissProt records, protein data bank, and other curated protein resources. So protein sequences are extremely important because protein perform most of the biological functions inside the living organism, and we all know that very well. So protein determine the biological structure and function. So let's start. First, open your browser and visit NCBI official website. Now go to protein databases. So from the database drop menu, select protein. And click on search. Okay. Now let's search for a well-known protein. So I will search for beta-lactamase. Okay. And click on search. Immediately NCBI returns thousands of protein records, and each record represents a protein sequence submitted or curated from various sources. You can see here beta-lactamase, the name of protein, what it shows, the page it shows beta-lactamase, that is the name of protein, the length of protein, that is 286 amino acid protein, then accession number. By using this number we can identify the particular protein, or if you want to submit any new protein you have isolated, so you will get that accession number. Then you can see here the FASTA format of that particular protein. The left panel, you can see here, the left panel contains useful filters that help narrow down your research. And these are, so according to source databases, so if you want your protein from PDB, that is protein data bank, so you can click here PDB. If you want RepSeq protein, you can select or filter that. UniProt KB also there, and some curated sequences are also there. Now NCBI allows filtering protein records by sources. Now let's open one protein record. We can click on RepSeq protein. I'm going to filter it according to the RepSeq. So suppose this is beta-lactamase protein, Apiospora phragmitis. I'm going to click on it. Now you can see here the protein that this is the whole information of that protein, that is beta lactamase isolated from Apiospora phragmitis, and here the accession number it is given, the source it is also given, organism, references, journal, everything it is given here. So if you can click here on protein, it will take you to the sequence of that protein. Regions are also given. Okay, and if you want to download FASTA format, just click on FASTA. Then go to send to button and click on file. So FASTA file, then create file. Your FASTA file get downloaded. If you open this file, so we all know that the proteins are represented with amino acids, and these codes, that is Mg, Q, I, Qg, so these are represented using a single letter code. So these are amino acid, that is methionine, alanine, aspartic acid likewise. In this way you can download the FASTA format. Now, so one of the most useful analysis available directly inside the NCBI is conserved domain detection. Okay. So we are going to see that. Okay. So see here, NCBI automatically compares the protein sequence against the conserved domain databases, and we can identify catalytic domain, binding domains, functional motifs, and evolutionary conserved regions. So if on the right hand side it is given, that is identify conserved domain, just click on it and it will take you to the another page. You can see here conserved domains. From here you can also find the similar domain architectures. Protein classification it is given here. Okay. Now the conserved domain databases and conserved domain search are integrated into NCBI protein resources for functional domain analysis. Now you can also BLAST the protein. If you go back and suppose you discover a new protein sequence and you want to know whether similar proteins are already exist, whichever you isolated the protein, whichever protein you isolated. So you want to search that if is there any similar protein is there which matches your sequence or your protein. So you can run the BLAST. So let's click on run BLAST. So why protein BLAST can be it can be used for sequence similarity searches, protein identification, evolutionary studies, and functional prediction. Protein BLAST compares protein sequences to protein databases and helps identify homologs and functional relationships. You can see here the accession number it is given of that particular protein, and if we just click on BLAST, it will take you to the protein similar to your isolated protein. Okay, it will take some time. Okay, now let's see here. Beta lactamase Apiospora phragmitis and it is also given here the E value. So this it matches only with beta lactamase Apiospora phragmitis. So because here query coverage it is 100% and E value it is 0.0, and percent identity it is also 100%. So remaining beta lactamases are somehow different from from this isolated protein, okay, which we are seeing here. Now let's see how we can do the advanced search. So let's learn some advanced searches that researchers used So for example, I'm going to search here again beta lactamase. So advanced field tags help retrieve highly specific protein records, and NCBI supports field specific searches such as title and organism filters for more precise protein retrieval. I'm going to type here beta lactamase. So if I want a beta lactamase particularly from Pseudomonas aeruginosa, so I am going to type here and Pseudomonas aeruginosa and just click on search. So it will show you only beta lactamases which are isolated from Pseudomonas aeruginosa. See here. So it finally narrow down your research. Then for example, if you want beta lactamase but heat shock, okay, so I am going to type here heat shock protein. We want heat shock beta lactamase. In the bracket and just click on search. Okay, so you can see here heat shock cryptosporium species and the protein. So likewise, if suppose you want kinase protein, or you simply can say enzyme, but it should be from bacteria only. So just type and write bacteria and just click on search. It will shows you only kinase protein isolated from bacteria. So this is how you can search the NCBI protein database. So you now know how to use the NCBI protein database like a researchers. So in this tutorial what we learn is protein search, protein record interpretation, then FASTA file download, conserved domain analysis, then protein to DNA sequence navigation and protein blast, and how to do the advanced search. So in the next video of this NCBI master series, we will learn the conserved domain database and conserved domain search tools in detail. So if you found this tutorial helpful, like the video, subscribe to the channel and share it with your friends and research colleagues. Thank you for watching.